Okay, so one for you, Dr. Plotkin. Do you think that the results that you found would apply to cell-free expression? For example, if GFP constructs were used to probe expression levels, were tested in an optimized cell-free protein expression system instead of in vivo, would you expect the same results?
If I have to go on record. So to the extent that initiation was obstructed and that was the primary mechanistic signal in our data set, I would expect that even in cell-free systems, initiation may be facing some obstructions. But I mean, there's nothing better to do than to do the experiment. We haven't expressed them in cell-free systems yet. We've seen very similar obstructions to initiation in yeast, at least, but a truly in vitro system we haven't tested yet. I expect that some of the laws of physics and some of the laws of ribosomal binding will obviously be the same even in cell-free systems.
Dr. Welch?
So we would have expected the same thing, and we actually have tested that for our polymerase genes. We've tested our entire polymerase variant set in a cell-free system, and what we see is a very different result. And this is a system where the tRNA complement is highly boosted. And what we see is actually a lot of the differences are gone, that all the genes are expressing at similar levels. And even things that were clearly poor in the E. coli are expressing pretty much just fine in the cell-free system. So that just goes to say more, emphasize more that our gene sets don't seem to be limited by initiation, because you would expect that ribosomes, the structure is going to form whether it's in a cell or in a cell-free system, and getting ribosomes on might be limiting. It could be that something else is limiting in the cell-free system, but the fact that we get actually even higher expression levels in the cell-free system versus in E. coli for some of the proteins suggests that something else is going on for at least these proteins.
Okay, Dr. Vogel, a question for you. There has generally been poor correlation between gene products predicted from gene expression and proteins identified by mass spec analysis. What are your thoughts on this, and are there any gene families or gene regions which tend to have better expression or identification at the protein level?
That's a very complex question.
You have 2 minutes.
To answer the first part, we've actually just written a little review on this, and we sat down and tried to accumulate all the data sets where we have protein and matching mRNA expression levels. And we tried to see a difference between the organismal groups, and there was nothing. The one and only trend, if you really want to see something in the data, was that the significance of the correlations increased with time. So basically, we're now at a stage where we can identify many proteins in mass spec experiments, up to several thousand. We can also quantify them, which is even more important, and we can quantify them reliably. So that makes the data better, and that improves the correlation between those two measures that is observed basically for any organism that you look at. The second question, groups of genes, yes, there are some functional biases in terms of protein versus mRNA ratios. Metabolic proteins tend to be expressed at higher levels, and they tend to have a higher protein per mRNA ratio compared to other functional groups. Regions of the genes or gene groups that tend to be detected more or less, that's a question that goes into a whole different area of like the challenges that we are still facing in mass spectrometry, for example, in terms of protein detections. Yes, mass spectrometry is biased towards, for example, more highly abundant proteins, but we still see a wide range of concentrations. We see about 6 orders of magnitude in concentrations in these proteins. So, yes and no. And the longer answer is in even more details, which is probably going beyond the 2 minutes.
Okay. So we are coming to the end of the hour, but I'm going to try sneak in a question or two. One of the final questions will be, what would be the characteristics to look at, other than CAI, to improve protein synthesis when working with recombinant proteins in E. coli? I'll start with Dr. Plotkin.
Well, I mean, I think that question gets to the essential fact that we do not, I think, despite these systematic experiments that have started to occur in different labs, we do not, nevertheless, yet have a principled way of answering that question. I think we really need more systematic and unbiased assays on different sets of proteins, even just in E. coli, to learn what are the rational rules guiding protein expression. And we need, on the one hand, hopefully to have assays that will teach us some simple mechanistic things that may be general, but they may not explain a huge amount of protein variation. On the other hand, have assays that are sort of elucidate more statistical black box, but highly accurate ways of predicting expression levels. And hopefully, both these approaches will come together at some point, and the statistical approaches will actually point to the underlying mechanisms. I think until that research is done, and I certainly think that's entirely doable in the near future, we can't really answer that question with any real confidence, unfortunately.
Mm-hmm. Yeah, I agree. I mean, I think maybe our guesses are better now with the data that we have, but we're still making guesses. We need to look at more proteins and more systems, really, to see if there's general principles. And it's a possibility that there won't be general principles, that the protein-specific factors may be so extreme that you have to do a separate study for every system and every protein. We'll see. We'll let the data tell us. And over the next couple of years, I think there's going to be a lot of data that points the way.
Mm-hmm. Great. Well, that seems a good place to end. More work needs to be done. So that'll keep you all employed for a while longer.
Thank you, Sean.
We're unfortunately out of time. I'd like to thank our panelists once again for being here today and for generously sharing their insights and expertise. Dr. Joshua Plotkin from University of Pennsylvania, Dr. Christine Vogel from the University of Texas, and finally Dr. Mark Welsh from DNA 2.0. Thank you also to our viewers for all of your excellent questions. We had many, many more than we could possibly answer, so I do apologize if we didn't get to yours. Please go to the URL at the bottom of your slide viewer now to learn more about products related to today's discussion, and look out for more webinars from Science available at www.sciencemag.org/webinar. Also, please share your thoughts about the webinar with us by sending an email to the address now up in your slide viewer, webinar@AAAS.org. Again, thank you to our panel, and thank you to DNA 2.0 for their kind sponsorship of this educational seminar. Goodbye. Thank you very much.
Thanks, Sean.
Great job.